Today, we're going to dive deep into a virus that has had a truly massive global impact, the human immunodeficiency virus, or HIV. It is, without a doubt, a master of disguise, deception, and biological sabotage. To really get a handle on HIV, we have to start with a pretty mind-bending question. How on earth does a virus manage to break one of biology's most fundamental rules and literally rewrite our own genetic code? The answer to that question reveals a strategy that's as rebellious as it is ingenious. You see, HIV belongs to this really unique and frankly fascinating family of viruses. They're called retroviruses. And that little prefix retro, well, that tells you everything you need to know about their signature move. Okay, so in biology, information normally flows just one way. Think of your DNA as the master blueprint, right? And RNA is like a temporary photocopy of that blueprint that you take to a job site. Information flows from the permanent DNA to the disposable RNA. But retroviruses, they completely flip that script. They have this special enzyme called reverse transcriptase, and it does something incredible. It turns their RNA, their temporary copy, back into a permanent strand of DNA. It literally works backward, which is where they get the name retro and their power to permanently change a host cell. So now let's zoom in on the virus particle itself, the virion. And I want you to look at this not as just some random blob, but as a precisely engineered machine. It's a tiny, sophisticated piece of equipment built for one purpose and one purpose only: infiltration. Every single piece you see here is a critical tool for a biological heist. Let's walk through this toolkit. First, on the outside, you've got this envelope. Now, this isn't just any envelope. It's actually a stolen piece of our own cell membrane, which makes for a brilliant disguise. And studded all over it are the viral keys, proteins called GP120 and GP41. Then, inside, you've got this cone-shaped core, which holds something called the P24 antigen. That's a really important marker we use for early diagnosis. And then at the very heart of it all, two identical copies of its RNA genome, so it's got a backup, plus those three absolutely essential enzymes that make the whole takeover possible: reverse transcriptase, integrase, and protease. All right, so with its toolkit ready, how does this thing actually pull off the heist? Well, it goes straight for the top. Its primary target is the command center of our entire immune system, a type of white blood cell called the CD4 helper T cell. The strategy is simple. If you take out the generals, the whole army crumbles. And this invasion isn't some chaotic attack. No, it is a meticulous, step-by-step process. As you can see here, it's a plan that takes it from the very first moment of contact all the way to turning our own cells into a factory for new viruses. Let's walk through it. The first move is all about getting through the door. In step 1, that GP120 protein on the virus, the key, locks perfectly onto a CD4 receptor on one of our T cells. But one lock isn't enough. It's like two-factor authentication. So for step 2, it also has to bind to a second co-receptor, something like CCR5. Only when both are engaged does that GP41 protein spring into action, basically harpooning the cell and fusing the virus to it. And bam, the virus is in. Immediately, its star enzyme, reverse transcriptase, starts doing its thing, rewriting the viral RNA into DNA. What happens next is honestly the most profound and frankly terrifying part of this entire process. The virus isn't just a temporary guest. It's about to make itself a permanent, hidden part of who we are. So how does it become that permanent time bomb? Well, that newly created viral DNA is marched into our cell's nucleus, and that's where the second key enzyme, integrase, does its job. It literally cuts our own chromosome and pastes the viral DNA right into our genetic code. This creates what's known as a provirus. From that moment on, our cell has been hijacked. Its own machinery is now forced to read this enemy blueprint and start churning out viral parts. Finally, the third enzyme, protease, acts like a pair of molecular scissors, snipping and assembling all those parts into new, fully functional viruses, which then bud off from the cell to start the cycle all over again. Now, here's the truly insidious part of HIV's strategy. This whole hostile takeover doesn't happen with a big bang. For years, sometimes a decade or more, a relentless war can be raging inside the body, all while the person on the outside feels perfectly healthy. The infection typically progresses through 3 main stages. After the virus gets its initial foothold, it enters a long period called clinical latency. And during this whole silent phase, which can last for years, the virus is anything but dormant. It's replicating like crazy, mostly in the lymph nodes, and it is slowly, methodically wiping out those crucial CD4 T cells. And this number right here, this is the tipping point. A healthy person has anywhere from about 500 to 1,500 of these CD4 cells in a tiny drop of blood. When that count drops below 200, the immune system is considered critically compromised. That is the clinical definition of AIDS. The command center has essentially been abandoned. And this brings us to a really, really important distinction we need to make. HIV is the virus. It's the pathogen itself. AIDS, on the other hand, acquired immune deficiency syndrome, isn't the virus. It's the diagnosis you get when the damage from the virus becomes so severe that your body can no longer defend itself. And this is what that vulnerability looks like. When the immune system is down, all these other germs and microbes that a healthy body would just brush off can become deadly invaders. We're talking about things like a fungus, candida, causing severe infections, or common viruses like herpes becoming rampant. In the end, what's so tragic is that for a person with untreated AIDS, the ultimate threat often isn't HIV itself, but one of these opportunistic infections that takes advantage of the body's defenseless state. But here's where the story takes a turn. By understanding every little cog and gear in this viral machine, scientists figured out its weak spots. They learned how to hack the hacker, developing an entire arsenal of drugs designed to jam the very enzymes the virus desperately needs to survive. The strategy is just brilliant. We attack the virus at every critical stage of its heist. For example, reverse transcriptase inhibitors block that first unique retroviral step. Protease inhibitors stop those molecular scissors from building new viruses. And then you have newer drugs like integrase inhibitors, which stop the virus from ever becoming part of our DNA in the first place, and fusion inhibitors that block it from even getting inside the cell. But the real breakthrough? It came from combining these weapons. It's a strategy called highly active antiretroviral therapy, or HART. It's a combination punch that uses a cocktail of 3 or more drugs at the same time. By attacking the virus from multiple angles at once, it becomes incredibly difficult for it to mutate and develop any kind of resistance. And the impact of this approach, it has been nothing short of miraculous. The development of HART has completely transformed the outlook for HIV. It turned what was almost certainly a death sentence into a manageable chronic illness for millions of people all over the world. It is truly one of the greatest success stories in modern medicine. But, and this is a big but, the fight isn't over, not by a long shot. These powerful treatments are amazing at suppressing the virus, but they don't eradicate it. Antiretroviral therapy is a lifelong commitment, not a cure. And there are still huge challenges ahead. And maybe the biggest challenge of all is the one that has stumped scientists for decades. After all this time, all this research, why don't we have a vaccine for HIV? Well, it turns out the virus is just a frustratingly brilliant opponent. There are 3 main hurdles. First, it's incredibly diverse. There are tons of different strains all over the world. Second, that reverse transcriptase enzyme, the one that makes it a retrovirus, it's really sloppy and makes a lot of errors. That means the virus mutates at a wild pace, so it's a constantly shape-shifting target. And finally, there's latency. The virus integrates itself into our DNA and can just hide out in these latent reservoirs, lying dormant for years, completely invisible to our immune system or to any vaccine we might develop. So I want to leave you with this final thought. The fight against HIV forced humanity to innovate in ways we never had before. It wasn't just about brilliant science. It required massive public health campaigns, tireless activism, and a global commitment to fight stigma and get treatment to the people who needed it. So, could this comprehensive, multi-layered response, this very blueprint that was forged in the fire of the AIDS crisis, be our greatest weapon for the global health crises that are still to come?